webinar, as I said, of our series, which we started in the month of June 5th, in fact, we started. But of course, in the month of July, we couldn't do that because of continuous lockdown in the city of Jorhat. But August, again, we have started. And this is the 9th webinar in our series. And today's speaker is very well-known scientist and, in fact, very beloved scientist, Dr. Sunil Kumar Mukherjee. Thank you, sir, for accepting our invitation. In fact, two times we had to postpone your webinar. For that, once again, I'm asking apologies, sir. Yeah. Okay, anyway, many of you, of course, know him very well because, of course, from our Assam Agricultural University, participants also, they know him very well because, many a times he visited our center, Northeast Center for Agricultural Biotechnology, because he served as a reviewer, external reviewer of our Scientific Advisory Committee. So, welcome you, sir, and then welcome all participants. So, but some of our new students may not be knowing him, so that is why a brief introduction. Dr. Mukherjee is now serving Indian Agricultural Research Institute as a senior INSA scientist, plant pathology. I think you joined, sir, about 3 years back, right, as a senior INSA scientist? Yes, 3 years back, yes. He completed his master's in physics. It's a very, kind of, transformation you see from physics to hardcore molecular biologist. So he did his master in physics from Vishabharati University in 1974, and then a post MSc in theoretical nuclear physics from Saha Institute of Nuclear Physics in 1976. Then he joined PhD degree in Calcutta University and obtained his degree in 1983 in biophysics. Then Dr. Mukherjee joined the biochemistry department of Johns Hopkins University, USA, as a postdoctoral assistant during 1983-84. And later on, he worked in the microbiology department of Duke University, USA for 6 years as research associate. Dr. Mukherjee has then he then started working in the field of DNA replication since 1984. And in 1990, he joined ICGEB, that is India International Center for Genetic Engineering and Biotechnology, New Delhi. And he started working in the P chloroplast DNA replication. And he established an in vitro system of replication in P. And then Dr. Mukherjee started working in the geminivirus. So geminiviral DNA replication, long years of experience he has in the geminivirus DNA replication. And he then started working with the artificial microRNAs, and then did the functional genomics in tomato, and then basically for tomato leaf curl viruses, to get resistance against tomato leaf curl viruses. So he is known to be a very well known expert in the field of RNAi technology. And he is a very good teacher also because he has visited numbers of organizations as visiting or guest faculty. And then he is a fellow of National Academy of Sciences India as well as Indian Academy of Sciences, and a recipient of Sri Ranjan Memorial Lecture Award from Nasi in 2008 and Pansanan Maheshwari Lecture Award in INSA 2010. So Dr. Mukherjee will speak today on RNAi, the Big Bang of Silence. So I welcome you once again, sir, and I'm leaving the floor to you. Thank you very much. Sir, please.
Thank you, Vidut, for your kind introduction. Actually, introduction is much bigger than my lecture. Am I audible, Vidut?
Yes, sir. Yes.
Okay. All right. So I have prepared this lecture in the form of a class lecture, and not a conference lecture, because I was told that MSc students and the PhD students will form the main audience. So this is just like a class lecture, okay? And here the silence means the RNAi, and Big Bang means its impact is like a Big Bang. So I have to tell you what the silence is, what's the mechanism, and its application, which works as a Big Bang. Okay. So as you see, that modern biology is very different from the classical biology, and there are actually pivotal points for this change in face of modern biology. First, the Watson-Crick's structure of DNA, and that explained how the phenomena of DNA replication, DNA transcription, and all other things can happen. So, it changed the genetics completely. And then, subsequently, the DNA sequencing and the genome sequencing, more than 200 eukaryotic genome has been sequenced. So, a vast knowledge is locked in the genome. Okay. Now you need to explore what is, how can you unlock the information presented in the various genomes. And there are two great techniques about it. One is RNAi, and the other.
Excuse me, sir.
Yes.
Sir, excuse me. Slides are not visible in a bigger form. I think, Dr. Modi, can you suggest or Rupjiti?
Sir, actually, individually, we have to search and pin the slides. The slides
Okay,
are visible
individually.
to
No,
everyone.
okay, that's fine. Yeah.
Individually, we have to search and pin the slides, sir.
Okay. How do I pin?
Sir,
Okay, pinning is, you have to, yes, I got it.
Mukherjee, sir, is so okay, sir.
Okay, sir. Sir, please go ahead.
Okay.
Go ahead, sir.
So there are two great techniques. One is RNAi and the other is CRISPR-Cas. And today's subject matter presentation is about RNAi. Before telling you what is RNAi, I wanted to impress the audience by showing what it can do for us. Like, a cow has been engineered using RNAi techniques, because the cow milk is very good for us, but for 1 to 2% of the consumers, it contains a protein which is beta-lactoglobulin, and which gives severe stomachache. So, in the mammary gland of cow, RNAi has been applied so that the milk is free of this protein. So, this kind of genetically engineered cow will give us milk which cannot give stomachache. Okay, this is. Let's come to another example, which is very, very recent. Application of RNAi as far as mammalian organisms are concerned is very difficult, because you have to apply RNAi at the very specific site. So the specific delivery is a very important point, and that has been achieved in the case of liver. Okay, and in the liver there is a protein called transthyretin, and it is just like a transport protein. It transports a vitamin and many other organisms. But due to some genetic defects or physiological defects, this protein is made in huge amount in the liver, and then it is transported to various tissues. Like if it is transported to heart, then it will deposit in the heart, and you will have a heart problem. If it deposits in the kidney, then you will have kidney problem, and so on and so forth. So one has to stop the formation of a huge amount of protein, and that has been done using RNAi. So you can apply RNAi medicine in the hand, and due to its site-specific delivery, the RNAi medicine will reach liver and will correct this disease. We have lots of antibiotics, we have lots of antifungal medicine, and many other antipathogen medicines. But virus, for virus, actually there are no medicines. To be very frank, there are two virus-specific medicines. One is herpes simplex virus DNA polymerase specific, and there is another one, I forget the name. So for virus, there is no general medicine, okay? But RNAi gives you a scope to stop viruses in a very, very specific manner. Like, the slide shows that there is a mouse which dies when it is affected by influenza virus. But if you treat the mouse with RNAi medicine and then apply influenza virus, nothing happens to the mouse. So there is a great antiviral medicine. Okay, so I thought, I'm talking about everything about mammals, so we should talk something about agriculture also. Recently in Latin America, RNAi has been applied in agriculture. It has applied in other places also quite some time ago, but this has been recently applied, and it is being mass cultivated. There is, as we eat rice, they eat bean. And bean is affected by a virus which is called bean golden mosaic virus. It destroys it completely. You will see that on the right-hand part of the panel. Okay, but if you treat this plant with RNAi-specific medicine, then the plant is completely unaffected by bean golden mosaic virus, as you see on the panel in the left. Okay, so these are something like, application aspects, which sounds like Big Bang. Now I'll come to the main topic. What is RNAi? On the top, as you see in the top border, there is a spectrum of life. Human being, gorilla, cow, snake, and then, snake and then plants and all those things. But this whole spectrum of eukaryotic life is controlled by a paradigm of law. And that is that every cell has their DNA, and DNA makes RNA, and RNA makes the protein. And the collection of proteins governs what the cell will do. Okay? So this is the general paradigm. But as you see, the biological life is not so simple. See, this linear array of activity doesn't always hold. RNA can be made from DNA, but that may not translate to protein at all. And sometimes the DNA to RNA transition is also affected. So these are all silencing mechanisms of which we'll talk about. Okay, it shows the same thing as I told you earlier, that DNA makes RNA, and then RNA is made non-functional so that there is no protein. Also, RNA could be degraded, completely degraded, by enzymatic means, not by anything. And this degraded part gets back to the nucleus and shuts up the transcription machinery. So the transcription is absent. So here you see the two types of silencing. The RNA is made, but protein cannot be made. This is called PTGS, post-transcriptional gene silencing. And over here, transcription cannot happen at all. And this is called transcriptional happening, transcriptional silencing. So the top part is called TGS, and the bottom part is called PTGS. All right. So some kind of PTGS is known in prokaryotic and eukaryotic biology since 1990 because of the discovery of the antisense RNA. Okay, but using sense RNA and stopping the transcription was completely unheard, and that is the topic of RNAi. Since 1990, this kind of silencing has been observed in plant. There are so many reports, and then C. elegans, the nematodes, and the fungi. There are plenty of examples. And this kind of things went on till 1998. I'll give you one example, one specific example of silencing in plant. So this is actually, I would say, the science of RNAi began from the discovery of Jorgensen's experiment. A company, floral company, gave the task to Jorgensen to increase the color of petunia. Petunia normally gives, bright red color, purple color, but they wanted to make it more bright so that they can sell it at a higher price to the market. So that was the idea. So they gave the job to Professor Jorgensen, and Jorgensen has seen that the color actually comes from a metabolite. And the metabolite is derived from a series of biochemical reactions. And the first panel of reaction is controlled by an enzyme called chalcone synthase. So he thought if we can overexpress chalcone synthase in the plant, then the color will be more bright. Meeting sorry. So that was the idea, and then he introduced the chalcone synthase gene from Medicago into petunia, and then wanted to look at the results. They thought the color, the flower color, would be more bright. In reality, some of the flowers were really more bright, but most of the flowers lost their color. In some, there is total loss. The whole flower was white, and in some cases there was sectorial loss. Some sector was white. So this is a strange observation because the transgene which has been introduced, that can go wrong, that can be silenced, but there is a resident gene that should give the color, but that is also not happening. So they named this phenomena as co-suppression. The transgene is silenced and the corresponding resident gene also is silenced because they have lots of homology. So this is one silencing phenomenon that I wanted to describe because this is the precursor from which all RNA experiments were derived. Later on, this kind of thing were found in many systems, C. elegans and others. So till 1998, people were in dilemma about not only the mechanism comes later, what is the exact trigger of silencing? You can silence by antisense. You can silence by sense also, sense oligonucleotide or sense genes. So what is at stake? What gives this silencing? So in the figure you see the two great scientists, Fire and Mello. They published two papers in 1998, and they were awarded Nobel Prize 8 years later for their discovery. And what they thought that they will prepare RNA from a specific gene of nematode, and that was one muscle gene called UNC22, UNC22. So they prepared the sense RNA, antisense RNA, and then mixed the two together to form double-stranded RNA. And then used chromatography to separate all these fractions separately so that you can have pure sense RNA of UNC22, pure antisense RNA of UNC22, and double-stranded RNA of UNC22. And fed these three different forms of RNA in nematodes. And they introduced in the head part of it. Okay. Now they wanted to see that which of the three types of RNA can silence the UNC22 gene of nematode. And if the UNC22 gene is silenced, then you will see a phenomenon called twitching. Twitching is that since the muscle gene is silenced, it wants to move, but it cannot move. So it takes this type of form, and it's called twitching. And twitching happened only in the case of introduction of very small amount of double-stranded RNA, which was not observed with antisense or sense RNA. But if you apply, 1,000 times molar excess of antisense RNA, then you can see some twitching. But here, a catalytic amount of double-stranded RNA delivered only in the head part can transform the whole organism in the twitched form. So not only the head part is twitched, the tail part is also twitched. Okay, so the UNC22 gene, wherever is present, from the head to bottom, everywhere it is silenced. And then they saw what happens to their progeny also. And they found that twitching phenomena also occurs at the F1 and F2 level. Okay, so then they repeated this experiment with many other genes of C. elegans. Okay, there are about 88 different genes they took, and in all cases, they got the similar result. So they were excited, and now they found one answer, that for silencing, double-stranded RNA has to be made within the cell. Otherwise, silencing cannot occur. Okay, and in the case of plant or C. elegans, a very catalytic amount of double-stranded RNA can silence the whole organism, actually. Okay, and now people have found that the silencing, same mechanism, that the creation of double-strandedness can give you RNAi-mediated silencing, and that happens not only in nematode, it happens in flower, it happens in human being, it happens in Drosophila, it happens in even yeast. There are two types of yeast. Cerevisia Saccharomyces pombe is competent of RNAi, but not the other form. Cerevisia doesn't give any RNAi. Okay, so if you see a silenced organism, then as we discussed earlier, you should expect that there will be double-stranded RNA corresponding to a gene at least. So from 1990 to 1998, there were many silenced organisms, especially silenced plants, and people wanted to find where are those double-stranded RNA. Okay, and I quote one of the great papers of Balcombe, who was not given Nobel Prize, but actually people think that he should have been given. The experiment is like this. He made transgenic tomato, and the transgene was ACO gene. And he took actually 5 transgenic tomato. And in the 3 transgenic tomato, transgenes are expressed very well. And for the other 2, no transgenes were made. So these 2 transgenic plants are actually silenced plants. Now they wanted to compare the RNA profile of these 5 different transgenic plants. And they found, ACO gene is about 1.1 kb long. So they expected 1.1 kb double-stranded RNA in their gel, but they couldn't find. On the top part, actually all the plant RNA looks same. But at the bottom, where the plants are silenced, there is a different signal. But that is a very low molecular weight, about 20 to 25 nucleotide long double-stranded RNA. So this actually was very surprising, because Fire's experiment told us that without double-stranded RNA, silencing cannot happen. But here you see the silenced plant where the double-stranded RNA is there, but a very low molecular weight. So how do you reconcile these two facts? Then it dawned to them that in the plants there are enzymes called dicerts, which cut the double-stranded RNA into small form. Small form means of 20 to 25 nucleotide long. Okay? And that was a seminal discovery, because later on people have shown that this small RNA, they're called siRNA, and they themselves are competent to cause silencing. So this is the example. These are human cells which are transformed with GFP. So in the normal phase microscope, you will see them like this, but in the in the UV illumination, you will see that the cells are illuminating with green color, okay, because they have GFP inserted inside. And now, if you introduce GFP-specific siRNA in these cells, the phase microscope gives the same view, but in the UV illumination, you see the GFP color is completely lost. It means the GFP siRNA can silence GFP gene. And if you give siRNA of different types, different sequence, nothing happens to the color. Now actually, well, this slide has come in a different form, but anyway, there is this sequencing technology which has been vastly improved. So small RNA or large RNA can be sequenced very, very quickly and with extreme precision. So you can sequence the small RNA which Belcom has seen, and they have found that the profile is split in 3 different forms. One is 21 more, the middle one is 22, and the other one is 24 more. 3 different siRNAs are formed. Okay. In other cases of silencing, you will get a bit longer siRNAs also, which is about 31. So this is one type of small RNA which is found in the silenced plants. And there is another type of silencing that always occurs in a living cell. And that is done by another small RNA, which is called microRNA. So in this presentation, we will talk less about microRNA, but more about small interfering RNA, that is siRNA. Let us come to the biochemical characteristics of siRNA. As we have seen that they are small size, the size is given between 21 to 24 mod. And at the 5' end, they have, since it is double-stranded, they have 2 5' and both of them have to be 5'. Both of them should contain 5' phosphate and 3' hydroxyl. Without that, they won't work. Double-stranded I have already told you. And they are not blunt-ended. They are actually staggered-ended, and the 3' end has protrusion. So there are 2 protrusions, one at 3' end on the top, and on the bottom end also 3' end. And the protruded ends are mostly U and U. You can have other nucleotides also. Mostly you have U and U. And these are the product of the enzyme which are called dicer. And after being formed, they form another riboprotein complex called RISC. And the RISC can actually degrade the mRNA, and that is silencing. Okay. Since this is a class lecture, I thought I'll tell the students how to make an siRNA. So this is, say, any sequence of an mRNA, and in the middle there is some sequence which are designated by N1, N2 up to N18 nucleotides. And I ask you to prepare an siRNA which will degrade this mRNA right at this site. So the way to do will be from N1 to N18, you add, and then you add 2 nucleotides which are U and U. And then for the other end, you make a complementary strand. For N18, if it is G, you have to put C here. So these are dash means the complementary nucleotides. And at the end, you put to another U and U. So that will be actually a siRNA. Now, if you add this siRNA with the mRNA and the protein complex, which is called a RISC, you will find the middle part of this sequence will be cut, completely sliced. As I told you earlier, that siRNAs are formed from the double-stranded RNA because of the presence of the enzymes which are called dicers. And dicers are organism-specific. Like in Arabidopsis, you have 4 dicers. Actually, they are called DCL1, DCL2, DCL3, and DCL4. Similarly, rice has 7 different dicers. And along with dicers, they have also RNase3 type of enzymes. But human being, for us, we have only 1 dicer. Okay? And you can recognize each dicer by its characteristics motif, which I will talk a bit later. And for its function, each dicer has to associate with its cognate RNA binding protein. For one particular dicer, there will be one RNA binding protein with which it will associate. For other dicer, this RNA binding protein, let's say you have 2 dicer, dicer 1 and dicer 2. Dicer 1 has RNA binding 1, and dicer 2 has RNA binding 2. And RNA binding 2 cannot work with dicer 1, so on and so forth. So these RNA binding proteins are extremely important in view. Okay, the motifs. You see, there are some conserved motifs of any dicer protein. Okay, there are so many dicer proteins known in the database, but if you look at them, two sequences are highly conserved. One is PAZ domain, and the other is the catalytic domain, which is called RNase3 domain. So these 3 domains are highly conserved. And in some dicer, you can have other motifs also, but not all. So these other motifs are not conserved, so to say. But in a subset, they are conserved. Like here, in a rice dicer 4, you have one RNA binding motif. Okay? In dicer 1 of Arabidopsis, we have 2 RNA binding motifs. But PAZ and 2 RNase3 motifs are conserved in any type of dicer. And now, the point is how the dicer attach to the RNA, double-stranded RNA, and produce the small-sized RNA, which are of the size, 20 to 24. To do that, actually, crystallography came in handy, and there is what is this called, Giardia. There is a parasite called Giardia, which has dicer, but its 3' end can be made easily soluble. They can produce a large amount, you can solubilize it, and then produce crystals. And when you produce crystals of the 3' end of the dicer, it looks like this. It looks like actually hook. The hook is at the bottom. And at the bottom, there is PAZ domain, using which it attaches to the RNA. And also on the top, you will see a boxed part. One helix is one RNase3, the other helix is other RNase3. There are 2 RNase3. So the 3-dimensional structure looks like this. And when you superimpose on double-stranded RNA, it will look like this. This kind of string are double-stranded RNA. And you see the end of the double-stranded RNA is attached by PAZ domain of the dicer. And over here there is catalytic domain of RNase3. So this catalytic domain will cut the double-stranded RNA here. So you will get a fragment which sizes from here up to here. And crystallographically, people have found that it is about 65 angstrom long. It means it can hold nucleotide of about 20 or something like that. Okay, and sometimes the distance is even more than that. It depends what is the structure of RNA and what is the structure of protein also. So from between 65 to 72 angstrom you get, and accordingly you get 21 more, 22 more, and 24 more. Okay, so the dicer makes siRNA. What happens later on? Once siRNA is formed, the siRNAs are bound with dicer. But then within the protein, within the cell there are proteins called arginate, AGO1. And once arginate is there, it attaches to the dicer, and then dicer leaves the place. And once dicer leaves the place, AGO1 protein brings in 7 or 8 other proteins of the cell. And together, they form a complex which is called RISC. And within the RISC, only one strand of the siRNA will be there, and that is called the guide strand, and the other strand will be degraded. So a RISC complex will form, which will contain one strand of siRNA. And that siRNA sequence will know where to attach with the mRNA because of Watson-Crick pairing. Now the RISC complex is actually a site-specific endonuclease. So it knows to which site of the mRNA it has to bind, and after binding, the AGO protein has the power to slice the mRNA. Okay, so after binding, the RISC complex will slice the mRNA. So I need to tell you about the AGO protein. Its three-dimensional structure looks like this, and it has also a PZ domain. It has also mid domain and PIWI domain. Its catalytic domain is actually a PIWI domain, using which it acts as a site-specific nuclease. So here is a model. It shows how AGO protein is complexed with siRNA and its cognate mRNA. The PZ domain of arginate protein is holding a yellow strand, and yellow is one strand of siRNA. Okay, and it forms a Watson-Crick base pairing with the mRNA here, and that's why that mRNA is here. And once there is there, the arginate protein undergoes allosteric change, and so that the PIWI motif of it is right in the middle of the local double strand formed between the siRNA and the mRNA. Okay, and then this PIWI domain has 3 amino acids together, and it comes together and cuts the mRNA only, not the siRNA, but the mRNA, right in the middle. So the mRNA is cut into 2 parts, and then that mRNA is destroyed by exonucleotic action of the cell. Now, putting together all these facts, we can talk about the mechanism of silencing. So somehow a double strand has to be formed within a cell, and then the double strand is cut by dicer along with its RNA binding cognate proteins. And once it is formed, this is called a diced complex. The diced complex form a complex which is called a RISC, and the RISC cuts the mRNA, and the mRNA is eventually degraded to nucleotides. And at this step, the silencing is total. It means silencing is complete. So you might think that that the basic mechanism is twofold. One is dicing, and the other is slicing. Slicing is this part. So in this way, you have dicer proteins and the RISC proteins. So you might think that it is the action of only a few proteins, but it is really not so. An experiment has been done in C. elegans, where starting from this step to final step, you require presence of 91 different types of proteins. Okay, so it all means that between dicing step and this slicing step, there are many sub-steps of biochemistry which is not yet understood. I've told you the silencing and also some silencing factors. Actually, amount of silencing factors present is organism specific. Like in Arabidopsis, you will find about 150. 150 means there are 4 dicer-like proteins, and there are 10 argonates, and there are many other RNA binding proteins, and so on and so forth. Altogether, about 150. Okay, in human being, you don't have that many. You have a smaller amount because human defense factor has others, like immunoglobulin and so on. So the silencing factors is organism specific. Now the question is, what is the biology of these silencing factors? Now that's a huge question, and actually I would say the information is gradually emerging. The information is not complete yet. Okay, so the first thing is that the silencing factor produce a small RNA, which I told earlier, which are called microRNA. Our organism development, and organism has disease also. So development and disease of organism is controlled by microRNA. So since I'm talking about microRNA, I have to talk very lightly about microRNA. I'll tell you how it is formed within the cell at the biogenesis. MicroRNA and microRNA and siRNA, they function almost similar manner, but their biogenetic pathway is very different. About 2% of the coding sequences, about 2% of the total coding sequences are filled by microRNA genes. And this coding sequence means some of them gives protein and many of them don't give protein. Only few give protein, 70% of them don't give any protein. That's 70% of our non-coding RNA. Okay, so a few small fraction gives miRNA genes, are occupied by the miRNA genes, and they are non-coding genes. So once they are transcribed by polymerase II, and also a small fraction of them are transcribed by polymerase III. Polymerase II, when they are transcribed, they form a precursor microRNA, which is called primary microRNA, and its structure is like this. So somewhere you will find a stem loop of type structure, and the 5' end there is single strand, and the 3' end there is single strand. And this will be then subjected to cutting of dicer within the cell. In the plant it is called DCL1, and then this stem loop structure will be cropped out. So this cutting will be called, cut product will be called another precursor microRNA, that's P microRNA. So pri microRNA is formed, and then after one cutting, you get P microRNA. And then P microRNA is diced further, which you, due to action of which you get only the stem part of it. And this stem part is actually microRNA, it contains microRNA and microRNA start. And then with presence of proteins like HT, they come into the cytoplasm, and after coming to the cytoplasm, they form Rix complex. Using the Rix complex, some mRNA can be clipped, and some mRNA can be made non-functional as far as protein making is concerned. And that is called translational repression. Translational repression doesn't happen with siRNA, but with siRNA, cleavage of mRNA happens, and miRNA can cleavage also. If the homology between the microRNA and the mRNA is very high, it will go in the cutting pathway. Okay, are you there with me? Vidutji?
Yes, sir. Yes,
Yes,
sir.
sir. Yes.
Yes.
All right, I'm moving on to my next slide.
Yes. Please.
In fact, if you have any problem, you can ask me questions meanwhile. I give you that license.
Okay. Thank you. Yeah, students can interact at any time.
See, I told you earlier that microRNA gave you the biogenetic pathway and function of microRNA. And I just wanted to show you that microRNA controls our genetic architecture. Like, we have 2 eyes only, not 3. And the eyes are below the forehead. Okay, so these are all genetic architecture, and these are all controlled by microRNA. In the case of plant, I'm showing you an Arabidopsis plant, which is in the juvenile phase, which has all its rosette leaves. And then when it transits to adult phase, a big shoot comes out. Okay, and then you have leaves also with them, and then eventually flowers are formed. So when the flowers are formed, it goes in the reproductive stage. So it has 3 stages: juvenile stage, adult stage, and reproductive stage. And all these stages are governed by microRNAs. That in juvenile phase is controlled by microRNA 159 and microRNA 172. And they give rise to another form of RNA, which is called siRNA, because of the action of the siRNAs, you get the juvenile phase. And if there is a mutation in this microRNA or the siRNA, then the juvenile phase quickly transits to adult phase. Okay, so to maintain the juvenile phase, you have to have this microRNA. Similarly, to have the flower reproductive stage, you have to have this kind of microRNA. And then again, you can ask the structure of the leaves. The leaves are not rounded, leaves are not narrow, elongated. Leaves have some particular morphology. And that morphology is maintained by, again, another set of microRNA, 319 and 396. And then roots. The roots are formed, they have different types of branching, and they are controlled again by microRNA 169 and 164. In short, that microRNAs control the development of the plant. There is another example from the maize plants. This is the juvenile phase or vegetative phase, and this is the reproductive phase. And the vegetative phase, microRNA 156 has to be low, and the reproductive phase, microRNA 172 has to be high, so on and so forth. Now the leaf structure, the sex formation, flower formation, etc., they're all governed by microRNA. And not only that, there are some microRNA which gives drought resistance, like MIR 166, MIR 169, and then from the soil it has to extract phosphate. So if the phosphate is low, then microRNA 133 comes to its rescue. So whole control of development is done by microRNAs. I won't say whole, but a huge part of it. I talked about development, but also the disease. You see, there is a virus called tobacco curly shoot virus, and this is Nicotiana benthamiana plant. And when this virus attacks, you will see the shoot part is getting curled. Okay, and then if you do the microRNA analysis, you will find that one microRNA, which is 1919 microRNA, which is actually depleted. Okay, now you can actually simulate this virus-like infection by depleting this particular microRNA by some novel technique. Okay, like there is a technique applied, it's called VIGS technique, using which the microRNA 1919 has been made down. And there is no virus, but still you get the same phenotype. You get the disease symptom. So microRNA also control the disease. Well, microRNA I talked about, but I wanted to focus on siRNAs only. You see, every organism has some genes which needs to be expressed during its development. And if it is expressed in a different time of development, then that is fatal. That is fatal to the organism. And thus, the genes needs to be controlled. And the silencing factor controls those things and give rise to the gene-specific siRNA, which are called endo-siRNAs. And then you have transposons. You are aware that, our whole DNA has about 50% transposon. So the transposon are very much problematic kind of stuff. They start jumping from one place to the other, and that way they can cause huge mutations. And if it happens, the organism's life will be at stake. So you need to control the transposons. The silencing factors control the transposons. There are many other things which I have described, but this part is most interesting. Silencing factors actually protects every organism, plant or animal, every organism from its invading virus. So it has a basic antiviral role. But this is interesting because if it stops the virus only, then we won't have any virus like coronavirus. We won't have virus like, there are many plant viruses. So viruses have also learned how to treat the silencing factors, and they are called RNAi suppressors. So the disease is actually an outcome of presence of this silencing factor and viral suppressing factors. If the suppressing factor wins, the virus will win, killing the organism. If the defense factors wins, the silencing factor wins, the virus will die. Okay, so I told you earlier that RNAi has evolved as an antiviral mechanism. It's a defense factor. But how does it defend? When the virus enters in a plant cell or any other cell, somehow double-stranded RNA is formed. There are many different ways of double-stranded RNA formation. In plant, it is formed in one way. In animal, it is formed in another way. In plant, also, it is formed in different manners. But anyway, double-stranded RNA corresponding to the viral transcripts are formed. Okay, they are mechanisms. And once they are formed, the double-stranded RNA is cut by the discs, and you get siRNAs from the viral genome, and they are called vsiRNAs. Then vsiRNAs actually form mixed complex, and they cut the viral transcripts. And if the viral transcripts are cut, viral protein will be formed. Okay, so in this way, this viral siRNAs protect the organism. And another form of protection, you will find a phenomenon which was known long ago, before the discovery of RNAi also. That is plant recovery. In fact, there are two phenomena. One is called plant cross-protection, and the other is plant recovery. We knew these things much earlier, before the discovery of RNA. Plant discovery is a thing that when plants enter, the siRNAs are formed, and the siRNAs move within the plant, okay, and they move in before the viral infection form can move. So in the emerging plant leaves, the siRNAs will be present, and when the virus comes, the viruses will be inhibited by these existing siRNAs, and that is plant recovery. Okay. And then the other half of the siRNA part comes in, that is the formation of siRNAs, or the silencing factors which make siRNAs are suppressed by viral factors which are called RNAi suppressors. I will come to that part a bit later. And as you understand, that if the silencing factors, if any of the silencing factors are mutated, then the mutated plant will be hyper susceptible to the virus. Okay, it shows different types of virus. I need not bother on that. And when you take flock house virus and allow them to enter Drosophila cell lines, you will find the viral siRNAs which are formed. Okay? And the discovery I've already mentioned, then I don't want to labor hard on this point. If there is any question, I'll come back to it. Okay. Now I come back to the RNAi suppressors. What do they do? Okay. RNAi suppressors actually protect the viruses. It's a counter strategy mechanism. It protects the viruses, and in its absence, some viruses cannot grow at all, and some viruses can grow, but they cannot create any disease. Okay? Here is an example. In the flockhouse virus has two genomes. One is a large genome and the other is a small genome. And within the small genome, they encode the RNAi suppressors. The large genome is capable of replicating itself. So to view the replication of flockhouse virus, the scientists have introduced GFP. Okay? So when the large genome is introduced in flockhouse virus in Drosophila cells, you will find cells which are infected with the RNA and they give fluorescence. So fluorescence means infected cells. Okay? Now, if your cells infect the wild-type Drosophila cells, then you don't see any colored dots at all. It means replication cannot happen at all. So, in the wild-type Drosophila cells has all RNAi silencing factors. And those silencing factors don't allow the flockhouse virus to grow. And that's how there is nothing. But in these wild-type cells, instead of adding the flockhouse virus large genome, if you introduce the silencing the RNAi suppressor itself in the form of plasmid, then you see all the cells are infected. Okay? Or rather, if you take AGO1 mutated line and then infect them with the large component of flockhouse virus, then also you can see these colored dots. And for these colored dots to come in, the RNAi suppressors, the homologous suppressors do very well. But the heterologous suppressors, that is suppressors derived from influenza virus or some other viruses, will also do. So the suppressors also act in a cross-kingdom manner. This is animal cell line example. I will give you a plant cell example. This is a Benthamiana plant, and a Benthamiana plant is killed completely by a virus which is called Cymbidium ring spot virus. Okay, Cymbidium ring spot virus has a RNAi suppressor which is called P19. Now, if you do a 1-point mutation so that the P19 cannot be made by this virus, and then you introduce that mutant virus, then you see that the viruses live very well. And you can do northern blots and others, you can find presence of the virus, but there is no disease. So, RNAi suppressors is actually helping component of the viruses, without which some viruses cannot live, and some viruses, they can live, but they cannot create disease. Some viruses have 3, some viruses have 4, some viruses have 1 suppressor, depending on the size of the genome. And this is an old slide. By now, RNAi suppressors are known many times. I guess about 70 or 75 plant RNAi suppressors are known. And in the case of animal virus suppressors, work has not been too much. About 10 are known. And in insect virus, about 5 are known. This is to show how a P19, the protein that I mentioned about some time ago, which comes from Cymbidium ring spot virus, how they act as an RNAi suppressor. See, this is an arginate protein. Normally, this is an siRNA, okay? This siRNA is held by Rix protein, arginate protein. And arginate protein holds the siRNA exactly in the same manner. And it is surprising that P19 protein also binds the siRNA exactly in the same manner. So when P19 binds to the siRNA, arginate protein cannot bind. And if arginate protein cannot bind, there cannot be any Rix complex, and there cannot be any RNA silence. I'll skip the slides. And this is something I mentioned you earlier, but again, I'm coming back to reinforce the point. I told you that if the plants are defective in the RNA silencing factors, then they will be susceptible to the viruses. Here we show different types of virus. In the first place, in the panel A, there is TMV virus. Okay, TMV virus is killing the tobacco plant. Sorry, on the left panel here, it is killing the tobacco plant. But if this tobacco plant already incorporates a silencing factor, which is known as RNA-dependent RNA polymerase 1, then nothing happens to the plant. Okay. Similarly, another virus, sun hemp mosaic virus, it kills the tobacco. But when the tobacco incorporates RNA-dependent RNA polymerase 1, nothing happens by the virus. So there are many other examples. Similarly, there are other examples. I'll skip these examples. And I would like to emphasize that animal viruses, especially coronavirus is very much in the top these days. Animal viruses are also protected that way. That is, animal viruses also code RNA suppressors to be pathogenic in human being. Okay, and the animal viruses has another layer of protection or deprotection. The plant has microRNA, and using microRNA, they can control viral gene expressions, and thus they can control viruses also. This phenomenon is not observed in plants. Okay, after this, I have slides on how to use small RNAs, siRNAs, how to make RNAi medicines. So before this, if you have any question to discuss, you are free to do it.
Okay, any questions up till now, whatever has been discussed? Sir, actually, sir, say miRNA, microRNAs are very small molecules, right?
Yes, yes.
Say, and then, so they may have, regulations for more than one kinds of mRNAs, say, encoded by different genes, right? So in that case,
it happens mostly in the case of animals. Animal microRNA controls many mRNA genes. By many, I mean more than 1,000.
Okay.
Okay. But in the case of plant, the scope is limited. MicroRNAs normally base pair very well with the mRNA. Okay.
Okay. Specific. Very specific.
Very specific. And that's how they cannot control many mRNA genes. But there are few microRNAs which do not match very well with the mRNA, and they control several genes.
Okay. Okay. Thank you. And then, suppose how do you validate this, microRNA, mRNA interactions?
Okay.
How this can be validated? Yes.
Yeah, actually that can be done because there are kits available for that. There are kits, animal cell line kits. Okay.
Mm-hmm.
So this kit is applicable for animal microRNA. But we can tailor-made it for plant microRNA also. It works like this. You have a cell line, okay, which expresses GFP. So you have the GFP encoding gene, and then the 3' end, you also have something. All right. And in the 3' end, so if you have this cell line alone, then it will fluoresce in presence of ultraviolet light. Okay, it will fluoresce green. Now in the 3' end, you put in a sequence which is binding region of the microRNA. All right. Now in that engineered cell line, you introduce your microRNA. So microRNA will bind at the complementary place, binding area, and after binding, because of the presence of the X complex, the GFP will be cut. So there will be loss of GFP.
Okay.
All right. So you take a particular sequence and you want to know whether it is interacting with the microRNA at all or not, you can use this kit.
Okay. Thank you. Because I had this, query. So anybody else is having any other queries, Hello, students. Do you have any questions? Otherwise, sir, please move to the next portion.
Good afternoon, sir. Can I ask?
Yes, please. Yes.
Sir, good afternoon, sir. Very happy to see you, sir.
Same here.
Yes, sir. I am Priyabrata, sir.
Priyabrata, good to see you.
Sir, what about the plant system validation? Transgenics is the way, or the cell line which you are talking about? And what about the experimental validation before going to transgenics?
Right. The experiment that I described now, you can use the same experiment. Okay. So the binding sequence has to be plant mRNA specific and mRNA, whatever you have, the conserved microRNA.
Okay. But for microRNA, mRNA base pairing, since I am also working little bit on microRNA and RNAi, as you know. But in the laboratory, we validate like with the technique like real-time PCR and RLM range.
No. Real-time PCR is no way to validate this. Real-time
regarding the
we'll give you the experiment.
Regarding
So actually, we discussed several times, and then I too suggest for more concrete validation. I discussed with Sam several times. So if you can give us some good guidance.
The validation that I talked to you about, is just a quick experiment type thing. But obviously, you have to create a transgenics to validate the thing.
Okay. Thank you, sir. Thank you, sir.
Most welcome.
Thank you, sir. Sir, please go ahead to the next slides, please.
What is the question?
miRNA will be synthesized in vitro? Question mark.
Yes, somebody is asking whether miRNA can be synthesized in vitro. The answer is yes. See, there are miRNA called artificial microRNAs, and all artificial microRNAs are synthesized in vitro. Most of them.
So in
Yes.
RNA, what is the role of siRNA?
See, siRNA has sequence using which it detects the mRNA. Okay, and once it is detected, the mRNA is destroyed. Okay, so I move to the next slides.
Yes, sir.
Okay, it shows that interest in the companies to show the various diseases that they can handle with RNAi. And actually, this is an old slide. Some of them do not work with RNA anymore, but a few more are coming in. But this company, Aninum, is a fantastic one, and then they are doing great discoveries as far as RNA medicine is concerned. The liver-specific medicine that I mentioned a few minutes ago are actually discovered by Aninum. Okay, so in a cell line, you can use siRNA and do anything you want. If you want to kill HIV, if you want to kill HPV, any virus you want, you can kill in cell lines. Okay, there is no problem if you can design the siRNA right. And then there are neurodegenerative diseases that can also be handled by siRNAs. Cancer can be handled, rheumatoid arthritis can be handled, autoimmune disease can be handled, etc., etc. And I'll give you one example like this. Gorilla, gets infected by Ebola viruses. You must have heard of Ebola viruses. Before this corona scare, the Ebola viruses were in the news, and the Ebola viruses are actually found within the gorillas of Congo River. And within 2 to 3 weeks of infection with the virus, the gorilla dies. But if you can pre-treat the gorilla with the siRNA specific to this Ebola virus, then nothing will happen to the gorilla. This paper came in Lancet in 2010. So in this way, actually, monkey level experiment has also been done at the Ebola virus. Monkey level experiment has been done in coronaviruses also. Okay, this Aninam group, I wanted to show they're doing great science. And there are another disease called RSV, respiratory syncytial virus. Especially it affects the children. This can be treated by siRNA, RNA medicine. And there is another list. There are many, many different lists. Okay, how we can make use of this siRNA technology to get to know what are the factors which are responsible for a virus infection. Let us say somebody is infected by influenza virus. How would you know that what are the host factors required for influenza virus? What has been done is like this. It has the Drosophila cell lines have been engineered in such a manner that they produce receptors to receive influenza virus. And then you introduce the influenza RNA components, and some of them you engineer with GFP for your observation's sake. Okay? So when this Drosophila cell line is infected with the influenza virus, you will find that the green color is coming up. But then within this cell line, if you keep on adding siRNA corresponding to Drosophila genome, okay, you take each genome, each gene, and then make siRNA corresponding to that, and feed with siRNA, and see whether the fluorescence is removed. If the fluorescence is removed, it means the virus is removed. So it means that particular siRNA which is coming from the gene, that particular gene is responsible for the influenza virus infection. In that way, you can identify the factors required for the virus infections. I move to plant biotechnology now. Okay, there are many different things that actually you can do. I'm not showing tables and others, but just mentioning few salient discoveries. See, there is a difference between animal protein and plant protein. Okay, because animal proteins are rich in lysine and methionine, but plant proteins are not. Although lentil and some others are a bit rich in methionine. So the question is how to increase the lysine content within the plant. So that has been, a dream thing from the time of Swaminathan and onwards. Swaminathan took the wheat lines, irradiated them with gamma rays, and it was reported that some of the lines have increased lysine content, which was later disproved. But then, using RNAi, you can make plants which are enriched in lysine. I'll come to that part a bit later. You can also use, nutritionally favorable plant foods where fatty acid synthesis can be lessened. You can make coffee where the caffeine part can be removed. You can make poppy plant, which has narcotic, you can make it free of narcotic. So many different things you can do. And then the slide shows that these are the target genes, and these are the effects, trait changes that you can do. For example, if you want to have a lot of grain in rice, then this gene will silence. Okay, so on and so forth. The slide is very long. So many things you can do in plant biotechnology. Not only biotechnology, aesthetics part. In the aesthetics part, RNAi also comes in handy. In Japan, the blue rose is extremely popular, and you can make blue rose from the red rose using RNAi. Now, I'm coming to the lysine part. See, in plants, it is not that lysines are not made. Lysines are made, but lysines are quickly metabolized to glutamate. And there is a protein, it's called LKR-SHD, which does the job. So if you can remove this protein, then lysine will be lysine will be over-accumulating in plants. Okay? And if you over-accumulate, then also there is a problem. Then there is an enzyme which makes lysine internally, it becomes inactive because of the feedback loop mechanism. So how to increase lysine? One way is that if you can do RNAi on this enzyme, or if you can do RNAi, or if you can mutate this DHPS enzyme. So that is the slide. It shows that if you do the RNAi on the enzyme, then the lysine content becomes 5 times higher. And if you mutate that another gene which makes lysine from this precursor, to mutate that, then also you can get about 2-fold higher. But if you combine these two, then the free lysine in the leaf is about 25-fold higher. So you can make lysine upregulation in the plants. But there is a problem. If you have lysine upregulated in the plant, the seeds will have it, and the seeds do not germinate well. So you cannot have very much higher amount of lysine content in the seeds. So you have to be satisfied with the 5-fold higher content of lysine accumulation, and that you can achieve either by removing the enzyme or making a knockout of that other enzyme, which is feedback loop sensitive. And not only this has been done in mustard, and this has been done in rice also. Okay. Then let me move to my field of interest, plant virus field. And to resist the plant virus, there is an easy technique. That is, within the system, you have to introduce a double-stranded RNA. And that you can make either by crossing or by transgenic means. Okay? You have a gene which is called, which I am mentioned here by BG. There is a particular viral gene. Any gene will do actually. But if you take the RNA suppressor gene, the effect will be better. Take any gene and make an inverted repeat of that, and within the spacing you give an intron. Okay, so this is the transgene from here to here. So this is actually an inverted repeat of the virus gene. Okay, so this is acting as a transgene. And if you make a transgenic plant with this transgene, then this transgenic plant will be resisting all viruses from which VG is derived. Here it is an example that in the plant, this is tobacco plant which is transgenic, and tobacco plant is infected with a virus which is called PVY, potato virus Y. Now, if you take the virus gene as a sense gene and make simply transgenic, then out of 27 transgenic plants, you will get 2 plants which are very much immune to PVY. Okay? And you get 2 plants which are resistant, but the rest 23 plants will be very much susceptible. If you take antisense part of that particular gene, you get 1 immune line and no resistant line, susceptible line after 4. Okay? But now if you cross these 2 transgenic, so in the one line, the hybrid line, you have both sense and antisense, then you find that the outstandard RNA is being formed, and now the number of immune lines shooting up to 10 out of 27. Okay? And the number of susceptible lines are dropping down. So in this way, if you can make a double stranded RNA corresponding to a viral gene, you can get a resistant plant. I will give a commercial application of that. Actually, when this transgenic was done, people didn't know that the effect is due to RNAi. But now we know for sure that the effect is for RNAi. Here, there is a virus which is called ring spot virus, papaya ring spot virus, which wiped out the papaya cultivation in Hawaii quite some time ago. And then a oat protein transgenic of this papaya plant has been made, and it is showing highly resistant properties. Okay. And here it shows that these are the transgenic lines, non-transgenic lines. Non-transgenic lines do not bear fruit at all. The transgenic lines have very healthy and they bear fruit, and they are sold in the market. And this transgenic fruit actually is being sold in the market for about a decade. So for 12 years, people in Hawaii are eating this papaya. If this papaya dose is not enough, then I will come to my virus of interest, which is tomato leaf curl virus. Okay. It is a very deadly virus of tomato. And Israel, I am told, there is no tomato which is free from this virus. And in India also, the virus is expanding in a similar situation. So, so we need something to resist this virus. But there are a lot of problems. Like this virus exists in multiple formats. It has many DNA genomes. But they have the similar disease phenotype. So this is a very difficult situation. It's like fighting Meghnad. Meghnad covers himself under cloud. So this is also a cloudy situation because you don't know which virus you are fighting, because there are many species of that virus. So fighting this virus is very, very, very difficult. And this virus is actually a geminivirus, and it is transmitted to the whitefly. So insecticide spraying help a bit, but it is again it destroys the ecology also. So it is not recommended. In tomato, there are some elements which are called TY, which offer partial resistance to the virus. So what we need, a broad spectrum virus resistance, okay? And that can be engineered, and that's what we did actually in our lab. All right. Here you see the, where is the first? Here you see the infected leaves. They're all hard because of tomato leaf curl virus. And it has happened because the white fly bearing the viruses have attacked. And if you extract the viral genome, you will find that they come in two formats. It has either DNA A and DNA B. These are all single-stranded DNA. And the size of the genome is very low, 2.7 kb. Either A or B, or A and a satellite DNA, which is called DNA beta. So they come in two formats, okay? And in some cases, it is highly devastating. So we have found, so this is actually a DBT project given to us to know what kind of tomato viruses are present. In our country, there are about 15 different species of tomato leaf curl virus, which devastate tomato field, starting from Cornell to Kashmir. And there are many companies, which fool us. They time to time come to us and say that they have a resistant variety. In fact, they have varieties with TY elements, which gives partial resistance. And even this partial resistance is season-specific. In some season you get it, in some other season you don't get it. So a broad-spectrum virus resistance is really called for the day. So what we did is this. We know that the small RNAs are siRNA and microRNA. And siRNAs, I have not mentioned you one phenomenon which is off-target space, off-target, which gives lots of off-target. If you made a transgenic corresponding to siRNA, only one, if you could make for one siRNA, it's fine. But normally people do a collection of siRNAs, the gene is long, then you get more off-target effects. So we thought instead of going by siRNA route, we will go by artificial microRNA route. We will make microRNA which will target the viral genes. And which genes? Which are RNAi suppressors. Okay? So that's what we thought. And to do this, actually we had to come up with technology to identify the viral suppressors. I'm not mentioning which technology used to know viral suppressors, because there is a long list of publications. And the result is that in our kitty right now, we have 40 to 46 different types of suppressors in our hand. So we have the suppressors. Now we choose suppressors and design artificial microRNAs to inactivate those suppressors. And to do that, we design artificial microRNAs, express them in plants, and then see whether they can resist the viruses. Okay. A bit more technical thing. I told you that we need a broad spectrum virus resistance. So we wanted to see that amongst the various different types of virus, various RNAi suppressors, there are 3 RNAi suppressors, AC4, AC2, and AV2. There are 3 RNAi suppressors, and obviously there will be many suppressor genes of various different species. So we wanted to look which sequence is conserved amongst all. Okay, and in that way we found that within AC4 about 27 more is conserved. Within AC2 about 35 more is conserved, and within AV2 about 32 more is conserved. Okay, and this conservation doesn't hold across all viruses, but most viruses. And we also carefully chosen this particular RNA suppressors. You see, if you design a mechanism using which you can inactivate this area, this will inactivate not only AC4, but will inactivate a gene embedded in it, which is AC1, which is a very much important gene for virus replication. So actually, you are designing a stone which can kill two birds at the same time. It can kill the AC4, it can kill AC1 also. Similarly for AV2, it can kill AV2 and AV1. And AC2 also, it can kill AC1 and AC2. So the design is a bit unique. And then we designed artificial microRNAs following some rules, which are published, so I have never heard of those. And once you get a conserved region, like this, AC4 conserved region, then you want to know which part of it gives the least off-target effect. For that, there is a web-based material. So we give this sequence to that website, and we find that the portion in green of this sequence gives the least off-target effect in tomato. Okay, so then designing artificial microRNA was very easy. We took this sequence and make antisense of that. Okay, instead of A, there is U, and so on and so forth. This is a microRNA, artificial microRNA we designed. And while designing, we also made one thing sure, that it doesn't give any more off-target effect. So its transitive effect is minimum. By that way, we have to introduce a mismatch at the T' end of the microRNA. Okay. Similarly, we designed AC2 microRNA, AC4 microRNA, AC2 microRNA, and AV2 microRNA. 3 different types of microRNA we designed and expressed them in plants. Okay, and chose the transgenic, which express these small RNAs. And these are the effects. If you have wild-type plants, you see the leaves are flat. Okay, wild-type tomato plant. But when you introduce the virus in it, you see the leaves are getting curled. The leaves are getting curled. But if you give the transgenic, there is no curling. The leaves are flat. So the transgenic, the AMIR transgenic of AC4 and AC2 can protect the plant very, very well. Similarly, we did for AC2 also, AV2 also. Here you see a blow-up thing. See, this is the wild-type plant, and this is the plant, wild-type plant, which is challenged with the virus. Okay, and this is the mock, which has been, so when it is challenged with the virus, you see its growth gets retarded. And if you make a blow-up, you see the leaves are crinkled, leaves are curled. Okay, but if you give the transgenics, the same virus, nothing happens. They grow as tall as this, they bear fruit and so on. And if you extract DNA to see whether the viral DNA is present, you will find none. So these are kind of immune lines, and we have advanced them to T4 generations. Okay, so this is another way, and we have alternative novel ways for virus resistance also. And I don't know how much time I have. Vidut, should I go?
Sir, how many slides you have?
I have few slides. So I will skip the alternative pathway, okay? I'll skip the alternative pathway because this also gives good effect. I'll skip those slides. And this is something which everybody is trying these days.
Yes.
What I mentioned to you is the transgenic pathway, but there are non-transgenic pathway also. That is, you have to make double-stranded RNA by some means. By transgenic way you can make, or you can make double-stranded RNA outside the plant leaves and introduce them in the plants. Okay, so this introduction could be in the linear form, could be in the hairpin form, could be in the siRNA form, and so on and so forth. You can introduce them by synthesizing in vitro, or you can produce them in bacteria. Okay. The problem is that when you introduce the double-stranded RNA in the plant leaf, they don't go into the cellular cytoplasm so easily. The entry is a difficult thing. So the solution is that you mix them with some peptide, self-penetrating peptide, or use other means. You bombard them with the air gun or give pressure so that the double stranded RNA can get introduced in the leaf cells, leaf cytoplasm. And the other problem is that they're not stable. Okay, double stranded RNA, once you produce outside and then spray on the leaves, they're not stable, they degrade. So there needs to be conjugated with a nanocarrier so that they are stable. And if you do that, then you have the desired things. So you can make double stranded RNA, siRNA, hairpin RNA, you can make in vitro, synthesize them in vitro, you can make them in bacteria, you can chemically synthesize them, and then spray this double stranded RNA in the plant leaves. Okay? And you can get desired effects. I will show that a little bit later. Or you can mix double stranded RNA with nanocarrier, clean nanosheets, or you can mix them with the peptides, self-penetrating peptides, and then mechanically inoculate them in the plants. Then these will also go, double stranded RNA will go within the plant cells. And once they go in the plant cells, then there are dicers, and dicer DCL4 will give 21 mods, DCL4 will cut it in 22 mods, and DCL3 will make 24 mods. Out of this, these 22 mods are very, very important because they move systemically within the whole plant. Okay? And this one gives DNA methylation, and both of them can degrade the mRNA. So you can make dsRNA corresponding to the plant gene. In that case, you will get plant phenotype. You will get you can make dsRNA corresponding to the viruses. Then you inject the virus, it will be virus resistant. You can give dsRNA corresponding to the fungal gene, and then add fungus, it will be fungally protected, and so on. Okay, so this slide shows that you can protect even beetles, you can protect pathogenic fungi, you can protect viruses, and viroids, and so on and so forth using application of double stranded RNA. Okay, so I conclude now. RNAi medicine are effectively siRNAs coupled with a couple of factors, silencing factors, and there are various forms of siRNAs about which I have not mentioned. And now the industries are coming up, and these are not only small industries. The industries like Monsanto, Bayer, they are multi-billion-dollar industries which are producing various different types of transgenic, antivirus, antifungal, anti-nematode plants, and double stranded RNA applications are coming up these days to replace the GMOs, the transgenic alternatives. I will conclude here and respond to the questions if you have any.
Thank you. Thank you very much, sir, for such an excellent talk, which is extremely useful for the students, not only for the students, even for the scientists working in the area of gene silencing.
But I hope
yeah, but I appreciate your energy also, because you talked almost for 2 hours now.
Over 2 hours. Okay. So
yes, almost. Thank you. Something is echoing.
Yeah.
Something is echoing. Why is it echoing?
Somebody saying something.
Yeah, Rupjiti, don't play now. Rupjiti. Hello.
Yes, sir. Someone is streaming in YouTube.
Why are you playing? Somebody is playing, the video perhaps, or the recorded version. We have the question answer, because there are a few questions.
I think it's from your
Okay.
Yes, sir.
Sir, there are some questions in the chat box.
Okay.
Ratna is asking, under stress, can hairpin produce different mature mRNAs, miRNAs?
It depends what kind of, it can. The short answer is it can.
Okay. So Mamta is asking, you can now leave the slides. Then, people will see you answering.
All right. How do I leave? Escape.
Now stop sharing the.
So I'll cut it.
Yes, sir. Thank you. So Mamta is asking, Mamta, can you ask yourself? Hello.
Manoj is
You are
here.
there. Yes.
Because you told me Manoj is absent. I can see Manoj.
Mamta is asking.
I'm here.
Yeah. I
No,
can
not
see
Manoj, not Manoj. Yeah, Manoj is here now.
I'm
Thank
here.
you for attending. Yeah.
Yeah, from the beginning.
Okay. Yes.
What is Mamta's question?
Yeah, in the text box it is there.
Yeah.
That mRNA can lead to translational repression or mRNA degradation. How the fate of mRNA is decided if it will undergo repression or degradation.
I have already, while discussing with you, I mentioned that part.
Yes.
mRNA has lot of match with the microRNA, then it will go in degradation pathway. But if the match is not very high, if the match is only in the seed area, then it will go in translational repression pathway.
Okay. Now, Debojit is asking, is there any possibility of crosstalk between RNAi and RNA editing pathway?
That's very interesting. Yes, there is, sure.
Okay. Then Molaya is asking, Molaya, can you ask yourself? Hello, Molaya.
Yes, sir.
You
Sir,
ask
thank you for
yourself.
the talk. I have several questions, but first is, are mRNA-based biomarkers reliable?
Not AMI-RNA. You are probably asking the microRNA-based
microRNA.
right?
Yes, microRNA-based biomarkers, yes.
Yes?
Yes, sir.
Yes, there are. But it has to be, this has to be secretory microRNA. Okay?
Yes, sir.
It has to be
Sir, and
microRNA, and to claim biomarker, you have to do lots of validation studies.
Yes.
and sir, what are the
sorry?
Yes, sir. So please carry on, sir.
For breast cancer, 3
Yes, sir.
microRNAs have been identified very well.
Yes, sir.
For diabetes also, I think microRNAs are known. And for other cancers, microRNAs are also known.
Thank you, sir.
And biomarkers.
So how about the regulations that are related to artificial microRNA or siRNA-based suppressors? What are the major regulatory concerns?
For
artificial microRNAs or siRNAs?
No, see, artificial microRNA you are expressing using a promoter. Okay, so all your regulatory mechanism will be put in the promoter that you are using. Whether it is sensitive, whether it is tissue specific, whatever. Okay, so for artificial microRNA, the regulation would be achieved by the promoter that you use.
No, I think she asked about the regulatory, not the regulation of the expression for the expression, but the regulation of policy regulation.
In terms of policy.
Policy regulation will be same as transgenics.
Policy is not my subject.
Yeah.
So, there are 2 more questions, if I'm allowed. What are the off-target issues? Is the off-target issue more in viruses and less in plants?
See, the question is, what is the gene first that you are using? Okay.
Yes, sir.
So, if you use a long gene, like viral rep protein, viral geminiviral rep protein, I mentioned that in pink golden mosaic virus, they used the siRNA of red protein, which is long. Okay. If there are many siRNAs, there will be many different off-target effects. But to minimize off-target effect, you have to have two different things. One is design, and the second most important thing is bioinformatics. If your bioinformatics are very good, then you can get rid of this off-target effect.
Okay. Yes, sir. So one last question. Is it possible to formulate an mRNA or siRNA-based spray against diseases? I mean, and will such formulations be effective if externally applied?
Yes, but I have a caution there.
Yes,
if you use siRNA,
yes, sir.
right, then
Yes, sir.
siRNAs which are rightly designed and 22 more, they will work much better than 21.
And why is that so, sir?
Because the mobility of 22 mods are much higher than the mobility of 21 mods.
Okay. Thank you, sir.
Okay. You will have spread somewhere, but you want it to be present within the plant everywhere. So
Yes.
your thing, whatever you are putting, has to be very much. 22 mods are much more mobile than 21 mods.
Thank you, sir.
Okay.
So, thank you very much. Is there any more question?
Sir, I have one question.
Okay, Manoj, please.
Is it pure?
Sir, why?
No, it's Manoj. They have the same voice frequency, same vocal frequency.
Sir,
Yes, sir.
why siRNA is better than artificial microRNA?
See, because if you design artificial microRNA, then for synthesis part of it, you need a lot of stuff. You have to artificially design it. Okay, you have to synthesize it. But the vector that we have created for the siRNA is very easy. There you can clone any gene of your interest, and then you can get a siRNA. So it is actually, methodology aspect. Hairpin is difficult to design, artificial microRNA is difficult to design, but not siRNA. Thank you.
Okay, Manoj. So, if there is no more question, then once again I thank Dr. Sunil Mukherjee, and I also thank all the participants for joining us in this very important webinar. And in this particular month, we may not be having any more because we are still trying to have one on 28, but that may not be possible. So maybe in the next month, we will meet again. Thank you once again, all of you.
Thanks, Vidya.
Bye-bye, sir. Bye, sir. Thank you.
Most welcome.